Okay. Hello, everyone. My name is Hunter Quon. I'm a postdoc at Arizona State University. I work in Dr. Carrie Hamilton's team. Unfortunately, Carrie couldn't be here this week, so you get me instead, um, which is fun because I'm going to present some of her work and Joanna's work, who also couldn't be here this week, and then some of my work related to the topic of microbial risks and considerations for reusing waste streams. Um, Unlike Becca Munich, I feel like I saw all the talks yesterday and so far today and thought, mine is a little maybe different and out there, but I'm unlike her, I'm not changing anything. You're going to get my work for better or for worse. And I feel like I'm going to talk about microbial risks, which have floated around a little bit in the last 2 days, but haven't been, I feel like, concretely presented on just yet. And so in that way, I kind of feel like I'm the bad guy here, and I'm going to talk about the scary stuff and the health implications. But go microbes. That's right. E. coli. So as I mentioned, I'm a postdoc. This is just a little bit about my background and my interests. I did my PhD at University of California, Irvine under Sunny Zhang, another microbiologist and virologist. And overall, I would say my research is vaguely in process modeling and risk assessment, mostly related to water sources, sustainable water sources, non-traditional waste streams, rainwater, things like that. And as I'm growing in my research, I'm getting more and more interested in community engagement and equity around water use and access. So as I said before, the word risk has come out a few times, more so today, of course, but I just wanted to really talk about it and define it. So risk analysis covers risk assessment, perception, management, government policy, all of those things in the context of risks as concerns to individuals, such as public health and microbial risk, for example, or chemical risk, but also to private sectors, organizations, and society at large, because risk, when studied and analyzed, kind of turns into policy. And then risk itself, I would define it as it's the probability or consequence and uncertainty of any adverse outcome. So we can talk about it from an ecological point of view, environmental, or down to the individual, such as the risk or probability of getting sick, which is really what I'm going to talk about today. So one of the most common frameworks in this field that I and many use is called the Quantitative Microbial Risk Assessment, or QMRA framework. So I put a little diagram here, sort of defining and showing each of the main steps of this framework. It's been around for several decades now. And really, you start with identifying what is your problem? What do you want to study? And then from that, what is your main hazard that you're worried about? And after that, then you get into the math. How are you going to mathematically model and define both the dose response relationship, which is the mathematical relationship or likelihood of, let's say, getting an infection after exposure to a certain pathogen. And then you also would need to define, model the scenario or the exposure. So when you conduct a QMRA risk assessment, it's good to be as specific as possible and really scope the literature for what's already been done before or what relationships have already been established. And then finally, you characterize your risk, which is usually a probability of an outcome. And finally, based on that outcome, you can provide management strategy recommendations. It's often based on empirical data or already established relationships, and modeling methods commonly used in this framework are the Monte Carlo simulation, which is really just a algorithm or technique to use a lot of repetition and iterations to try to get your probability or outcome as confident as you can. And before I do that, I kind of wanted to more recently get into the more chemical risk side of things. So I just overlaid this exact version, but I changed a few words. I changed the word pathogen to contaminant, infection to outcome, to say that when you conduct risk assessment, even though QMRA by definition is used for microbial risk assessment, the general framework is very similar if you want to conduct chemical risk. If you know your hazard in your situation, and maybe the EPA already has an established relationship between, let's say, a carcinogenic metal and a slope factor for cancer, that's not too different from a dose response assessment. So I would say that the general flow of the framework is similar no matter what you are studying and working with. So this is where I open up the can of worms and say that I'm mainly talking about today antibiotic resistance or antimicrobial resistance and wastewater. So I know this conference is about fertilizers and waste streams and recycling. So I'm going to say that for me, I'm focusing on wastewater treatment as my waste stream. So about half of my presentation will be about looking at treated wastewater effluent reuse related to agriculture, and also the treatments and stabilization and reuse of biosolids as fertilizer for agriculture. And in recent years, wastewater treatment trains and plants have been identified as hotspots for enriching AMR or antibiotic resistance. And that is because of secondary treatment or activated sludge basins, for example. They're highly dense, lots of bacteria in there doing their thing, treating the water. But we also talked about a little bit yesterday that pharmaceuticals get in our wastewater from our urine. Everything is in this wastewater treatment plant, and it just becomes a nice place for bacteria to transfer their genes if they're already resistant, for example, and then when they're exposed to pharmaceuticals, the resistance spreads. And applications of reclaimed wastewater, there's a variety of them. So agricultural irrigation, what we're talking about today, but also urban reuse and even irrigation for non-agricultural settings. So we talked about golf courses yesterday, or just fountains, for example. If you see big ornamental fountains, sometimes they use reclaimed wastewater for those. Also cooling towers and toilet flushing. So all of these could be potential uses of reclaimed wastewater, therefore possible exposures to contaminants. So the first example I'm going to work through is a QMRA that I did in the last year on reclaimed wastewater for agricultural application. So just an example of how QMRA works. I collaborated with a team from Spain, and based on their study and their data, we decided to focus mostly on E. coli for this study because fecal coliforms are regulated in both unrestricted urban reuse of reclaimed wastewater and food crop irrigation with reclaimed wastewater. And it's commonly studied in risk assessments in general, so there's already pre-established relationships with dose response. We modeled the exposure through consumption of produce that has been irrigated with reclaimed wastewater. And then once we move through to the next step, we can take the exposure dose and model the mathematical relationship between this dose and, like I said before, the probability of infection. In this case, we did the unique focus or spin on it of trying to account for antibiotic resistant bacteria, which has become more and more a hot topic in this area. And of course, it's really difficult to model. Are you with me so far? Am I? Okay, cool. So I told myself maybe this was the change that I made last night that I wasn't going to. I thought I'm going to avoid using equations as much as possible. And then last night I had this just, I don't know, because I wanted to. I just wanted to say briefly that dose response, this portion of QMRA, when you've done it enough, it's actually, in my opinion, pretty simple. Generally, I'm not going to get too far. There's only two equations that you get to really pick from when doing dose response, because clinical data and clinical relationships, a lot of them have already been modeled and fitted by other people, and there's a nice database out there called the QMRA Wiki. So like for E. coli, you can kind of just go look it up and find the nice parameter and use it yourself. So if you kind of know how these two equations work, you just pick the one that has the better fit. So the first one is the exponential model, has the D, which is the number of pathogens or the dose, and the only other parameter is R, which is just the best fit parameter for the exposure model, and it really just assumes that all the pathogens have the same survival probability, and that's what R is. So if you have a number for R, you have the whole equation right there. And then there's the beta Poisson model, which has two different parameters. So that's it. I just wanted to show those out there and say those are the two equations that we work with in QMRA in general. But recently, in my last group at UC Irvine, a guy who was mostly studying computational biology and he's a whiz at math, developed a spin on the dose response assessment to account for and include antibiotic resistant bacteria. So his motivation was try to keep it as close to these two equations as possible, but add an extra piece to account for both resistant and susceptible populations. So without getting too much into the details, I'll say that the foundation of it is called the birth-death process or the simple death process. And essentially what he did is take that R value and make a few more assumptions about it and split it into two populations within the dose so that each resistant or susceptible to antibiotic population has a different R value. Also included in the model are assumptions regarding background or residual antibiotics. So we talked about before about pharmaceuticals being in your urine. If you take antibiotics or you were taking them a week ago, you may have trace amounts of antibiotics in you already. And then if you get exposed to a resistant population, there's going to be some unique interactions between those residual trace antibiotics and the dose itself. So believe me, they're accounted for in the equation, at least in somewhat. I won't get too much into the details though. So I mentioned before I collaborated with a team in Spain. They had a really nice data set they sampled several greenhouses, which I learned very quickly are a massive operation in Spain. And the ones that we studied were all growing tomatoes. They were all using reclaimed wastewater and drip irrigation. So they collected samples on the tomatoes from the irrigators themselves and from the soil. And then they came to us and said, well, we have data on antibiotic resistant bacteria. We want to conduct risk assessment and see if there's anything we need to be worried about. Each of the three greenhouses we looked at for the data was a little unique in how they handle their water. For example, greenhouse 1 just irrigates directly from the distribution line from the reclaimed wastewater plant. Greenhouse 2 brings the water in and then stores it in an open reservoir for about a month before it enters the irrigation system. And then greenhouse 3 mixes on site 3-to-1 with groundwater. So here's just some cumulative fractions of the data. And using Monte Carlo and fitting some distributions, I can pull out 10,000 theoretical samples from within the limited data set that I have. And all I'm going to really say about this is that the only interesting takeaway is that for greenhouse 1, most of their data were non-detect, which is a good thing. So I'll get into that when I show those results right here. Greenhouse 1 essentially had no risk. When I ran it through the model 10,000 times, the numbers were less than 10 to the minus 14. And like I said, most of them were non-detect, so that makes sense. We're basically saying there's 0 E. coli, so the answer is 0. But even with greenhouses 2 and 3, the risk was very low, less than 10 to the minus 10, so it's very low. And I didn't show it on here, but at least with the EPA, it's commonly used 10 to the minus 4 as sort of like a benchmark. If you're above 10 to the minus 4 or 1 in 10,000 infections per 10,000 people, that's when you maybe want to say, okay, hang on here, that there's maybe a concern, maybe we should look into this some more. And we're well below that, so that's a good thing. But also what I plotted here was I plotted the median risk for these given scenarios, which again is consumption of the tomatoes that have been irrigated, so raw tomatoes. But within the confidence interval, I went from black to red. So this model, I would say, was probably the first, I believe it was the first time we were able to use QMRA to have a unique outcome to account for resistant bacteria. So as you see, when you increase the background residual antibiotic concentration from 0 to anything above 0, you'll transition from black to red depending on the data set. So that all that is saying is that if a person were to be exposed to that dose and that fraction of resistant bacteria, if it's red, it means that that infection is likely to be resistant to that given modeled antibiotic. And that's really what we're worried about. And then briefly, we also, from them, had soil data for Pseudomonas. So with a separate exposure model, Pseudomonas is more known for causing eye and ear infections. So instead of consumption to tomatoes, we modeled contact with the soil. So this is more of maybe an on-site worker type of an exposure scenario. The risk was definitely higher, maybe not quite problematic just yet. It's at antibiotic 0, it's right at 10 to the minus 4, so a little questionable, but we still see the same trend from black to red, which makes sense. It's expected given the model. This study and another study where I use the same model, but in a sort of a different spin, were recently published. And I would say that the main outcomes of both are, given the results, the non-trivial risk and the new red outcome, kind of leads us to the question of from a policy standpoint, should we be considering resistant bacteria as maybe a new threshold or a new regulation in reclaimed wastewater and agricultural samples around this policy in this area? I would say probably yes, especially in the coming decades. We're going to see more and more resistant bacteria in these settings. So what about biosolids and fertilizers? I promised I would talk about that a little bit. So now we're going to look at the bottom half. And I already said that wastewater treatment and the secondary treatment is like that hotspot for resistant bacteria. But of course, those solids have to go somewhere, which means those bacteria have to go somewhere. Maybe, hopefully, we stabilize the sludge, which means we're essentially treating it and reducing the bacterial counts. But at the end of the day, just like the water, it's probably going to be used for agricultural application. So everything I'm presenting after this is a lot of Joanna Harrison's PhD work. So if you have any questions at all, email her, don't ask me. But ultimately, building from the bottom up, from here to here, she is looking at stabilization through agriculture application and then what that ultimately means for risk. So the first part of her PhD, I believe, was she did this massive review of reductions due to sludge stabilization of pathogens, antibiotic-resistant genes, and some other contaminants. I'm only presenting here the results of the antibiotic resistance genes. As you can see, there were a lot more data points. So the top one is for all of the reviewed studies that use an activated sludge system, and the bottom is for all of the suspended growth systems. And then along the way, there are all the different classes of antibiotic resistance genes. Tetracycline, heavily studied, but also one of the main ones that has negative reductions in stabilization, so a lot more variability there. But there's also, across the review, a lot of variability in site conditions, treatment type, location, not a whole lot of representation in low and middle income countries, for example. So I guess one of the annoying takeaways of this is there needs to be better standardization and reporting of units when talking about reductions of a treatment in this case, and also more representation across the globe of different treatment types. I've also seen the word trade-offs get thrown around a little bit, and I would say that that is probably the main goal of what Joanna is trying to do in her PhD is look at biosolids, look at different waste streams and identify the trade-offs, but with a risk sort of spin on it. So we've seen the many applications, benefits of waste streams, nutrients, sustainability in the last 2 days. But I would say from a risk perspective, what I like to do research on is there still needs to be a better understanding of quantification in general and reporting of all of these data points for someone like me to take and move them into a model like this. And if we want to do anything comparative, going beyond just E. coli, maybe we want to look at PFAS, for example. I'm not going to do that, but PFAS or chemical risk or E. coli, not just fecal coliforms, but other bacteria. There's data is definitely lacking in all of those to be integrated within a risk model, I would say. Julia presented a good chunk of this yesterday, so questions go to Julia. But Joanna and Julia studied the lettuce growth. These are more results from the lettuce growth experiment. So CAP water from Arizona, which is the Central Arizona Project, is aqueduct water coming from the Colorado River, and it often gets recharged with treated reclaimed wastewater. So we're kind of back to the reclaimed water paradigm and using a bunch of different combinations and scenarios of waste as fertilizers. We have some results of growth studies. Here are some of the preliminary results of Joanna's analysis. I'm only presenting the pathogens because that's really what I'm focusing on today. Luckily, a lot of them weren't even detected, which is great in the lettuce, on the lettuce, in the soil, and even in the water. But after the growth, the E. coli, which is the one plotted here, more or less found higher concentrations or higher counts in the manure and the biosolids, followed by lettuce, final soil, and water. So it wasn't even detected in the water. So I would say maybe the takeaway here is if it's not in the water, but we're irrigating the lettuce and we're still finding it in the lettuce at the end of the day, there's probably some sort of transfer going on from the manure and the biosolids to the lettuce throughout the duration of the period of growth. So that's all that to say is, even if we are treating our water really well, if we're using biosolids, there could be some cross-contamination or exposure to workers and consumers that we may not think about. And this is still ongoing. So this is just a very preliminary table of the overall results of that study, just of all of the different contaminants from the soil, the lettuce, and the water, and what we may want to focus on from a trade-off point of view. So I already mentioned that in the baseline, which means in the manure or the biosolids by themselves, we see the pathogens, but then we're seeing in the lettuce afterwards. So that may mean transfer from the baseline treatment into the soil and the lettuce, and leads us to wonder, should we quantify the risk from consumption or contact to the soil? And I would say yes. And Joanna agrees. And that's going to be I would say, the last big part of her PhD, which is where I stole this diagram from. All this to say that we're looking at, or she's looking at, multiple waste streams. So urine, manure, biosolids, and reclaimed wastewater, using all of them in irrigation for agriculture. Going to do a lot of math and a lot of data accounting, and then move it into a risk model to do a trade-off analysis. And, oh, sorry. My last little piece of me that I want to say is, a really useful metric for doing this when you're expanding beyond just microbial risks and probability is to use an entirely new metric that you can use across chemical risks, cancer, and microbes all the same. And it's called the DALY, or the Disability Adjusted Life Years. It's essentially a metric to account for how much a disease may impact your life at the end of the day. If you get cancer and you unfortunately would pass away 10 years early, you would have 10 years of life lost, for example. And the cool thing about, not really cool, but the interesting thing about that is you can sort of take the DALY, the amount of life impacted or lost, and move that into an economic assessment. We can put an economic value to the statistical human life, or to a disability, for example. So then we can say that we can give a cost loss due to life, due to microbial infection, due to cancer, due to any of these contaminants. But then we can also compare that cost on the other side to the benefits of all of these waste streams. How much money can we make using manure and urine in agriculture, and kind of trying to find the nice balance between, hopefully there's no risk, hopefully we don't have outbreaks and get people sick, and we are improving agriculture at large. This was my last slide. I just wanted to throw in, this is more of what I'm doing right now. I talked about those equations at the very beginning and how there's a lot of data missing, and it's really challenging to do this work for antimicrobial resistance. And one of the missing pieces, I think, is conjugation rates. That is, at what rate are bacteria transferring these genes to each other in different settings, in your gut, in wastewater treatment plants. So I have this under review. I did a massive literature review to try to find all the people that studied rates and in different media, in different wastewater treatment settings. And nothing really interesting to show here other than they're all over the place, but we need to understand this better as well from a modeling point of view. I want to acknowledge my advisor at UC Irvine, Sunny Zhang, and Samira, who was my collaborator in Spain and her team, as well as everybody at ASU, and Julia and Clinton as well. Thank you very much.